Um, Good morning, everyone. So, um, thanks for this kind invitation and for having me here. I'd like to share with you some of the work that we're doing on creating new kinds of microscopes, new kinds of sensors and diagnostic tools. And in my title, I also have an emphasis to democratization because I believe some of these designs that we're working on and others in the field are pushing forward will help us uh, democratize the landscape of advanced measurement tools that Vivek also was mentioning throughout his talk. There's a huge need for some of these lab instruments that we find in traditional hospitals or point of care offices in advanced settings. They should also work in the most resource poor environments, even in field settings in some cases. And that's where I think some of the work that I'll share with you is leading to. Um, in addition to field, in addition to resource poor settings, there's also the angle for the home. Can you convert the home into a 24/7 lab so that you can manage chronic patients, aging populations right at the home through their bathroom, living room, bedrooms? I think that's also coming, and there's some great evidence that these kinds of technologies can be translated even to the home, to the consumer. At the center of a lot of these developments, uh, we're leveraging consumer devices, specifically mobile phones. Mobile phones are special in many ways. Smartphones are special in many ways. And we make use of them for creating new things attached to them. Before I share with you what kinds of things you can do with a smartphone enabled device, I'd like to share with you some interesting observations regarding mobile phones and how they have emerged. In this slide, what you see is essentially the optical camera interface of your smartphone has been following the Moore's law. Its megapixel has been doubling every 2 years for the last 10 years almost. The red dots are the megapixel counts. This means the optical camera interface of a mobile phone has advanced exponentially. And you can actually build very sophisticated optical sensors and microscopes attached to mobile phones to the level where, with the recent 40 megapixel mobile phones that we have in some of our smartphones, you can actually see individual viruses. A virus is about 100 nanometers. If you divide your human hair by 100 and then divide that by another 10, it's about a virus. So you can see individual viruses, and even a single DNA molecule can now be seen. I'll share examples of that. A microscope that is essentially converted from a mobile phone, a handheld device, that can see an individual DNA molecule. That's now enabled through these kinds of rapid progress that consumer devices, in particular mobile phones, enabled. In addition to the optical hardware, these devices are also supercomputers. They have graphics processing units so that you can capture a raw image or a sensor data, but that at the same time process it rapidly using the mobile phone. And these are, I think, extremely exciting opportunities put together. And all of this is in its current form factor because of the volume. We have about 7 billion cell phone subscribers, and this is the reason why your iPhone is still a few $100s. And of course, they now work everywhere, which means this could be a platform, very advanced, very sophisticated, that you can push to even the most, again, resource poor settings. There was a great emphasis about sub-Saharan Africa and the needs in global health and all these resource poor settings. I think this is a great platform to push some of these advanced technologies even to the poorest places because of the widespread use of mobile phones. This is exactly what we're trying to do. We're trying to create new mobile health telemedicine technologies that leverage this infrastructure enabled by consumer devices, mobile phones, and components of these mobile phones, like CMOS imagers, the silicon chips that you capture digital pictures with. So what you see on this slide are some of these devices that in the past we've created attached to mobile phones. All of them are 3D printed, and that's a great advantage because then you can close the cycle of these designs with rapid prototyping. Within just a single day, you can come up with the third generation even tried out. Now it's routine to convert your mobile phone into a microscope, as I said, to the level where you can see individual nanoparticles, viruses, and even a single DNA. If you do not want to modify your phone, the CMOS imager, the silicon chip that is part of your optical camera interface on a mobile phone, can be taken out, and you can create actually standalone devices. So these are miniaturized and cost-effective interfaces, but they're also different in the way that they work. The physics of these devices are different. I'll give you examples of how they work very briefly. But for example, this microscope that I'm pointing to can generate 1 billion useful pixels. It's a gigapixel microscopic imager enabled by computation and components borrowed from mobile phones. In addition to microscopy, which is very general, you can also do other types of instrumentation around the mobile phone, including detection of biomarkers like albumin in urine, or you can create a flow cytometer. It can capture a video sequence, image sequence, and process it within a microfluidic device to give you essentially count of cells that are specifically labeled and are flowing through your volume. In this slide, I'm sharing some other interfaces. You can actually combine the power of your microscopic imaging system that you can attach to a mobile phone in fluorescence, in bright field, and write applications that can actually process these images for specific tasks. For example, in this embodiment, you have a blood analyzer. It can count red blood cell densities, white blood cell densities, and hemoglobin density in whole blood samples, all within less than half an hour, including the labeling time and all the sample preparation, with about 5 to 6% accuracy compared to a hematology analyzer. So in this regard, yes, there is some drift, some coefficient of variation, but these are tolerable, especially considering the handheld and extremely cost-effective and rapid nature of these tests compared to standard lab routines. Some of the examples are shown here. You can now detect bacteria, E. coli, Giardia, crypto, specifically and sensitively using interfaces attached to mobile phones. This is a recent work which uses nanotechnology, nanoparticles, plasmonic particles, to look at color changes in response to heavy metals. In this case, you're looking at mercury 2 ions in drinking water, and it has reached to a level where it can sense 3 to 4 parts per billion level of contamination, nanograms per mil. So in that regard, really, these are showing you some examples where sensitivity, specificity that you expect from a lab instrument can now be pushed to the field through these kinds of interfaces. Mobile phones are special for many reasons, as I've explained, but there are other emerging technologies like smartwatches or variable computers, especially glass is a great example. We've also written one of the first applications for biomedical use of glass. In this case, you're looking at a reader for rapid diagnostic tests. These are lateral flow tests, some of which are quantified, yes, some of which are just qualitative, yes or no, but some of which are quantitative. So a glass camera interface together with server processing can be used to quantify in a hands-free and voice-activated interface these tests in an easy way. And we've done some other work with glass because it's, I think, unique in many ways. However, the sensitivity and the computational power of glass and many other variable computers are nowhere near the smartphone yet. So in that regard, a lot of these interfaces use the cloud as a means to support the computational needs of some of these sensors and imaging systems. That's something to keep in mind. These are valuable not just because they're cost-effective, compact, accurate, sensitive, and field portable. They're also exciting because this is a network of measurement tools. So you're forming essentially a network which you can spatially and temporally tag these results and do analysis on them for epidemiology, for looking at trends and cause-effect relationships. And I think that's a very exciting area where this connectivity will help us reveal emerging patterns that are normally lost because we do not always record or connect this data sets that we have at different parts of the world. So in the next few minutes, I'd like to give you some examples. This was a brief overview, but now I'd like to give you some more examples of what you can do and how competitive some of these interfaces can get. And I'll start with microscopy because microscopy is universal. It can be used for various different tasks. So some of these interfaces that you see here are not your father's microscopes in terms of their design. They're actually quite sophisticated because there are some advanced algorithms behind them to make up for the lack of complexity in their design. For example, this microscope attached to a mobile phone, this one, or these devices that utilize a CMOS imager taken off a camera phone, do not have any lenses in them. They're lensless, they're optics free. All the bulky optical components, including objectives, objective lenses, that you normally find in a microscope design are taken out. So we don't have that sophistication that you find in a normal microscope. However, we have algorithms to make up for that. These designs are lens-free. That's why they're very compact and cost-effective. We've got rid of a lot of these expensive alignment-requiring designs and replaced them with essentially computational algorithms to bring you an interface that is high throughput, that has high resolution, that has high sensitivity to the level where it can literally see individual viruses, as I've said, in a field-portable way. So because it doesn't have any optics in it, literally, no lenses, there's no image formation. You deal with only shadows. These are imagers that literally bring the sample very close to the sensor. So this rectangular box here, you can think of it as the CMOS silicon chip that is borrowed from a camera, a webcam or a cell phone camera or a tablet camera, whatever you have. If you take that silicon chip, which is roughly speaking 0.5 centimeter by 0.5 centimeter, you place a sample of interest to you. It can be a tissue sample, it can be a microfluidic chip that has captured certain cells of interest, and you shine light onto it so that you capture shadows and sample, digitize them with this simple optics-free shadow imaging system, exactly like Xeroxing. But computationally, it's very rich. It gives you the basis to actually reconstruct images that exactly look like a traditional light microscope. At the raw image, for example, if you look at whole blood, you'll be seeing shadows of these red blood cells, sometimes white blood cells. It can work like a cytometer to count cells, but it's more advanced than that because these shadows are actually holograms. They contain useful information which can be inverted computationally. A field of view like this within a few milliseconds can be reconstructed using Fourier optics tricks to give you images of the cells at their plane, creating these shadows, nicely matching to a conventional microscope image. Except that there is no microscope, there's no optics. So it becomes very powerful. There's some nice physics behind this. I'll skip this, and you can always refer to my website and find these reference articles to go about it. This is not traditional holography. It's actually not even using lasers. So it's incoherent or partially coherent holography. It can be very powerful in a very simple 45-gram microscope. This is a simple LED here. The sample goes from the side. It's USB powered. There's a CMOS imager that is, in this case, 5 megapixel. And it can give you images that, in this simple design, may be about a 10x objective lens, which means you're looking at a resolution of about 1 micron to 2 microns, a modest resolution. But one major advantage here is the field of view. These are large field microscopes. Large area samples can be seen with these microscopes because these are unit magnification designs where the chip's area is your field of view. So this is a huge advantage because traditional light microscopy is typically a millimeter square in field of view. So you have an advantage here, but resolution is modest. That's something that you can expect from a 10x objective lens, as I've said. But you can actually push the resolution of this system to diffraction limit and mimic higher magnification, higher resolution imaging systems with some complication. Maybe from an initial design as shown here, less than 50 grams, very inexpensive to put together, something a little bit taller, but still less than 150 grams. So it's a field portable, very lightweight microscope, but it has a little bit more sophistication to it to improve the resolution without sacrificing field of view. If you look inside, what you see here is actually a bunch of LEDs. The sample goes from side. You capture holograms or shadows as before, but now the light source shifts. It gives us the framework to synthesize higher resolution images using a mathematical theory called pixel super resolution. Another way of saying it is we capture slightly jittered images and put them together exactly like what you see in this movie to synthesize a higher resolution image. Some security cameras use this kind of technology to reveal your car plate numbers if you're moving fast with respect to the camera system. It's the same thing that we do now computationally to push the resolution of this system to the diffraction limit. So using a field portable system that has this kind of technology, everything is computational and the hardware is still very inexpensive and simple to put together. In fact, this prototype that you see here, also 3D printed, has been put together by essentially an undergraduate researcher. So it's at that level where even relatively poorly trained students could put them together. Of course, there's a postdoc behind this kind of work. But you can see high-resolution images. In this case, you're looking at a blood smear, thin blood smear for malaria diagnosis. These are malaria parasites, Giemsa stain that invade a red blood cell, and this is the same thing that you can see in a lens-free microscope, without, again, the bulky microscope. One important advantage of this kind of a design is that it has a very large field of view and high resolution at the same time, so that it has more space bandwidth product, more number of pixels compared to a traditional light microscope. If you were to look at a state-of-the-art CMOS, for example, taken from some of your iPhones, you're looking at an active area like this. This is the main ingredient of the phone. It's so small that we put a sense image next to it. This is the area across which you can do imaging, and it's about 20 millimeters square. Very large for microscopy purposes. It can give you about 1 billion pixels. There are 1 billion pixels here that you can resolve about 200 nanometers feature size. This is another design which brings color to this system, so it's super resolution, color, and lens-free microscopy put together in another embodiment that is also put together by an undergraduate student. It's very sophisticated. It can take images, raw data that looks like this and quickly process it to give you, in this case, these are histopathologist slides that show you subcellular features across a very large field of view. This is one of our images. If you zoom in, you would see our reconstructions, but traditional microscope would be only looking at these kinds of fields of view. In terms of features of interest for a pathologist, we've given these images blindly to an expert pathologist, board-certified pathologist, for diagnostic purposes, and we've achieved 99% accuracy compared to traditional light microscope, revealing that all the features are preserved. That one error was actually entirely a statistical error of the diagnostician, not our images. We're very confident on that. So this is another example. It's a pap smear. The advantage is also shown here. These are our reconstructions, traditional light microscope, large field of view, and field portability compared to a traditional light microscope are the advantages, especially for practice of telemedicine. Now you can actually capture images and create three-dimensional microscopic movies, as you can see here, focusing to different parts of the specimen, and a pathologist a few hundred kilometers away can actually look at these movies and go back and forth to different planes to make essentially diagnosis feasible, even if you don't have the presence of the expert. More sophisticated designs, still field portable and cost-effective in implementation, can give you even virus-level information at the sub-100 nanometer level. This is actually a design which can see about, smallest particle that it can see is about 40 nanometers across a very large field of view. So this is something that you can also start to use as a nanoparticle sizing tool or a virus quantification tool if you have the surface chemistry or a microchip that brings you specificity. And it also not only detects these particles, but also counts them and sizes them with about a sizing accuracy of 10 nanometers, all the way from a single nanoparticle per 20 millimeters square per 10 microliters up to a few 100,000 particles in a dynamic range that is huge. You can actually start to do a quantified nanoscale analysis on your specimen. Could be quite useful not only for biomedical purposes, for viral load measurements, in the case, for example, of HIV-positive patients, but also could be applied to air quality monitoring. Imagine these devices deployed because of their cost-effectiveness in hospitals, in schools, in metro stations, to dynamically quantify the distribution of particles in air, all the way, again, from sub 50 nanometers to a few tens of microns dynamically. You can also bring some additional functions to this platform. It's a platform technology. I'm sure some of you must have heard of BioCore's surface plasmon resonance technology for protein assays, for DNA or protein binding assays. You can bring the same kind of technology by merging plasmonic apertures with this kind of an embodiment so that you can have a reader for surface plasmon-based assays, very similar to these expensive and bulky instruments. It works with similar transmission shadow imaging based approaches to give you these binding kinetics, this time at the molecular level. So we're now detecting monolayer formation on a surface functionalized gold nano aperture array, and you can functionalize it for different DNA or different proteins specifically, so that you can understand within a complex specimen the amount of binding to different regions in the form of a microarray. But the readout here comes in a very fast and obviously cost-effective and field-portable implementation. So I want to come back to this slide. That's where I started. A lot of what I've shown is literally riding on this curve, rapidly improving its performance every 2 years, maybe doubling its number of pixels, thanks to you and other consumers using more frequently, changing your phones, your consumer devices. That's the drive for the industry to come up with new designs. We're leveraging it and creating designs or microscopes or sensors that literally improve their performance as fast as this logarithmic curve. This is probably the first time that microscopy is really enjoying Moore's law because of the design simplicity here. Without changing much, with the same set of algorithms, we can actually enjoy a 40 megapixel imager. Compared to previous years where we used to have 10 megapixels, we can drastically improve our performance. Now I'd like to also share with you what we've done with some of these recent generation smartphones that had 40 plus megapixels at the back of their camera interfaces, especially for fluorescence, especially for fluorescence sensing. So far I've been more focused to bright field, to shadows, to diffraction, to seeing natural contrast, endogenous contrast. But obviously fluorescence is extremely exciting because it has some additional contrast mechanism that could be quite valuable, especially if you don't have a replacement in the form of natural contrast coming from the sample. Using some of these smartphones that have large megapixels at the back, we've created fluorescence microscopes using 3D printing to convert the phone into an advanced imager. In this case, you're looking at a dark field and fluorescence microscope, also 3D printed, which has sensitivity to see viruses and nanoparticles, sub-100 nanometer particles, very nicely matching to traditional light microscopes in terms of sensitivity. This is a great example where we're comparing our cell phone image, where these green dots are individual viruses, human cytomegaloviruses. These are viruses that have surface proteins, glycoproteins that are labeled with fluorophores. Maybe 500 to 1,000 fluorophores per spherical virus particle. That's what is imaged per particle per virus in this cell phone design. And now our comparison is a confocal microscope to really validate that we can image individual viruses. This can be a great way of measuring viral load in a complex specimen by different surface chemistries. Recently, we pushed this even further and started to see stretched DNA molecules. You can take a sample, a droplet that contains a target DNA molecule, and by stretching it between two cover slips, you can actually randomly linearize these double-stranded DNA molecules. So it's a very simple few minutes of sample prep, which will essentially, on a cover slip, create lines of these DNA molecules, which you can then label with these intercalating dyes to have them fluoresce. And essentially, this is a cell phone image. This is a high-end oil immersion objective lens comparison of the same field of view, and everything that you see here as these sticks, these are individual DNA molecules that are about 2 nanometers in width, and they have maybe 5 to 10 fluorophores per base pair that is giving you this fluorescence signal. And it's quantified using machine learning, using server-based processing. You can capture these nice images off a mobile phone, send them to the cloud, and have analysis on them to reveal the length. Our length measurement accuracy with this cloud-based mobile microscopy platform is about 1 kilobase pair for 10 kilobase and longer DNA molecules. In this regard, you can also use this kind of an analysis for looking at copy number variations. One thing that we're pushing this for is understanding drug resistance in malaria and TB by looking at the count of certain genes within the backbone of the DNA. Now you can start to do some of these genetic experiments using a microscopy tool that can see and size different genes specifically through labeling. Same kind of infrastructure, which is cell phones interface with some additional optics and an application and the server backhand to give you machine learning power. Could be used for specific identification of various different parasites. In this case, you're looking at a design that can look at Giardia particles, Giardia cysts, waterborne cysts in drinking water with about 1 cyst per mil level of sensitivity. It's the same idea. You capture these raw images, which are then sent to a server, which has learned already how the signatures of these Giardia cysts look. It counts automatically, kind of a self-contained system, giving the results back to this Windows-based phone for the user to quantify. And finally, before I conclude, this is a very recent work which has converted the phone into a microplate reader. These 96 well plates are one of the most commonly used platforms for bringing sensitive measurements to clinical settings in high throughput, so that you can actually have thousands of patients screened in a reasonable amount of time. But the readers for these 96 well plates are usually expensive and maybe a large printer-sized device, all the way from $20,000 to $50,000. What we created here is a ELISA test reader for 96 well plates that has machine learning at the server back end and some 3D printed design, which uses the cell phone itself to read all this entire 96 well plate colorimetric changes rapidly. This is something that we've also tested with about 1,100 patients at UCLA Clinical Microbiology Lab with mumps, with measles, HSV1 and HSV2. These are for IgG tests for immunization. So we believe that this kind of an interface could be quite useful for vaccination campaigns, especially in field settings, in point-of-care settings. Bringing an advanced tool like ELISA reader to the palm of the person in the form of a handheld mobile device. And our results were pretty nice with machine learning. It was about 99% accurate compared to an FDA-approved ELISA reader. To sum up, we believe that computation, when combined some of these opportunities created by mobile phones and other consumer devices and variable interfaces, they create a unique interface and unique applications for us to push some of these advanced tests, advanced sensing platforms, microscopy platforms, diagnostic platforms that we normally enjoy in labs to field locations, to resource-poor settings, for global health, obviously, but at the same time for also bringing some of these to the home, to convert the home into a 24/7 laboratory for spatial tracking of chronic patients. I also have to disclose a conflict of interest. I've started up a company that's based in Los Angeles, which is commercializing some of these technologies. And this is actually one of our first product, which is a rapid diagnostic test reader for different types of lateral flow of tests. And it's a universal reader which can work with almost any test that you have. With this, I'd like to conclude and thank you very much.